Okay, let's begin our third chapter on the circulatory system. Now we'll talk about the blood vessels and circulation. In this chapter, we'll begin with the general anatomy of the blood vessels, then we'll look at blood pressure, resistance, and flow, how capillary exchange is accomplished, that is, nutrients are exchanged at the tissues, and then venous return, as well as circulatory shock. We'll look at the anatomy of all these vessels towards the end of this chapter. We'll consider the pulmonary circuit, the systemic vessels of the axial region, as well as the systemic vessels of the appendicular region. To begin with, arteries will always carry blood away from the heart. Now, in the systemic circuit, that means the arteries are carrying oxygenated blood away from the heart. When we look in the pulmonary circuit, we'll see that arteries are carrying deoxygenated blood away from the heart to the lungs, and then the veins, the pulmonary veins, carry oxygenated blood back to the heart. However, in the systemic circuit, arteries will always be carrying oxygenated blood away from the heart, and veins deoxygenated blood back to the heart. There's difference in structures in arteries and veins, and we'll elucidate those here soon. Capillaries are the very smallest vessels. They connect the smallest arteries to the veins and allow for the return of blood to the heart. There are three basic layers of all vessels. They're called tunics. We'll have the Tunica Interna or the Tunica Intima. We have the Tunica Media, and then the Tunica Externa or the Tunica Adventitia. The tunica interna is the internal layer that lines the blood vessel. It's the layer that's exposed to the blood. The endothelium of it is simple squamous epithelium, which overlies the basement membrane and a sparse layer of loose connective tissue. It acts as a selectively permeable barrier, and it secretes chemicals that stimulate the dilation or constriction of the vessel. Normally, it repels blood cells and platelets to prevent clots from forming. The tunica media consists of a smooth muscle layer. It's collagen and elastic tissue. It strengthens the vessel and prevents the blood pressure from rupturing them. These muscles can contract against a volume of blood in the vessels. Vasomotion is the change in diameter of blood vessel, and it's brought about by the smooth muscle of this tunica interna. You can see both vessels have this layer, however it differs in composition slightly. The tunica externa, or the tunica adventitia, is a layer of loose connective tissue that often merges with the neighboring blood vessels, nerves, or other organs. It anchors the vessel and provides passage for small nerves and lymphatic vessels. The vasa vasorum are small vessels that supply blood at least to the outer half of the large vessels, because they can't get blood from diffusion out to that distance. Blood from the lumen is thought to nourish the inner half of the vessel by diffusion. But these vasa vasorum that we see right here will supply the outer 1/3. This image shows the structure of each of the vessels that we'll examine in this semester. We'll look at both large veins, medium veins, and venules, as well as large arteries, medium arteries, and arterioles. Let's begin by looking at the various arteries. The arteries are sometimes called resistance vessels because they have relatively strong and resilient tissue structure that resists the high blood pressure exerted by the heart. The conducting, elastic or large arteries, such as the aorta, common carotid, subclavian, pulmonary trunk, and common iliac arteries, are the largest of the arteries. They have a layer of elastic tissue called the internal elastic lamina. It's right at the border between the interna and the media. It's almost indistinguishable from the elastic fibers of the tunica media. There's also an external elastic lamina at the border between the tunica media and the tunica externa, but it too is sometimes imperceptible. These vessels expand during systole and recoil during diastole, which lessens the fluctuation in blood pressure. So when the heart pumps and pushes a bolus of blood out into the main arteries, they'll expand and contract back against that pressure. So it's almost as if the vessels have a pumping action themselves. Now, the distributing or muscular arteries are medium in size. They're sort of like off-ramps to the major highways of the conducting vessels. These distribute blood to all the specific organs. They're vessels like the brachial arteries, femoral arteries, renal arteries, and splenic arteries. The smooth muscle layers constitute 3/4 of the wall's thickness. Now, the resistance vessels are much smaller. They control the amount of blood to the various organs. They have a thicker tunica media in proportion to their lumen. And the arterioles are the smallest of these resistance arteries, with very little tunica externa at all. Meta-arterioles are very short vessels that link the arterioles to the capillaries. The meta-arterioles often have precapillary sphincters at the entrance to the capillaries. These sphincters are made of muscle cells, and they reduce or shut off blood flow to their respective capillaries when blood is not needed in that region, so we can divert it to other tissues. For example, when we're exercising, we need to have the capillary beds full in our muscles in the legs. However, when we're resting and digesting, those capillary beds will be shut off by the precapillary sphincters, and blood will be allowed to supply the digestive organs. So in the arteries, there are various sense organs. These are structures in the walls of certain vessels that monitor things like blood pressure and chemistry. They transmit information to the brainstem, and that serves to regulate the heart rate, vasomotion, as well as respiration. The carotid sinuses are the first ones we'll look at. They're baroreceptors, or pressure receptors. They're on the walls of the internal carotid artery, and they monitor blood pressure. You can see the carotid sinuses pictured here. In response to high blood pressure, the carotid sinus baroreceptors will decrease the heart rate and dilate vessels in response to high blood pressure. The carotid bodies are chemoreceptors. They're oval bodies that are located in the carotid artery. They monitor the blood's chemistry. They mainly transmit signals to the brainstem and the respiratory centers and adjust the rate in order to stabilize the pH, the carbon dioxide level, and oxygen levels in the blood. They're very much more sensitive to the carbon dioxide levels than anything else. The aortic bodies are also chemoreceptors right in the aortic arch. They function in the same way as the carotid bodies, by measuring the chemistry of the blood. Now, it makes sense that these receptors would be in these particular spaces. The carotid bodies are there right before the blood enters the brain. We're making sure that it's perfect. And also right before the blood leaves into the system in the aortic arch. So capillaries are a site where nutrients, wastes, and hormones pass between the blood and tissues through the walls of the vessels. These capillaries are composed of endothelium and basal lamina. Because of the density of capillaries in tissue, it's rare that a cell is more than 6 widths away from a capillary. The exceptions to this are in tendons, ligaments, epithelia, and the cornea of the lens of the eye. There are 3 types of capillaries that we'll look at here. First of all, continuous capillaries. These are the capillaries that occur in most tissues. Their endothelial cells have tight junctions forming a continuous tube here. The intercellular clefts between the endothelial cells allow passage of small solutes such as glucose. You can see some of the intercellular clefts right here. They also have pericytes, cells that surround them. They contain some contractile protein and muscles, and they're thought to be able to regulate blood flow to some degree. They're also involved in tissue repair because they can differentiate into both endothelial and smooth muscle cells. We'll also see fenestrated capillaries. These are most prevalent in the kidneys and small intestine. They have little windows, fenestrations. So the endothelial cells themselves actually have holes all the way through them. These are called filtration pores. They're spanned by a very thin glycoprotein layer. It allows rapid passage of small molecules through. We'll see fenestrated capillaries mostly in organs that require rapid absorption or filtration of substances. Sinusoids are discontinuous capillaries. They're sort of a dead end. We find them in the liver, bone marrow, and the spleen. They're irregular blood-filled spaces with large fenestrations. This allows proteins such as albumin and clotting factors and new blood cells to enter circulation. We'll examine each of these types of capillaries as we move throughout the semester and investigate various organ systems. Capillaries are organized into networks called capillary beds. The meta-arteriole, as we've already explored, is sort of a thoroughfare channel between the arterioles and the venules. The precapillary sphincters are involved in controlling which capillary beds are perfused. When the sphincters are open, we'll see that capillaries are perfused. When they're closed, we'll see that the capillaries are not perfused. About 3/4 of the body's capillaries are shut down at any given time. We just don't need to supply every single organ with the full amount of blood at all times. So the veins are regarded as capacitance vessels. Because they are relatively thin-walled and flaccid, they expand easily to accommodate increased blood volume. If we look in this figure to the right here, we see that at any given moment, the veins contain more blood than any other vessel in the body. They have much less muscular and elastic tissue because they're not subjected to the pumping of the heart. The blood flow through the veins is relatively steady. Small venules will combine to form larger veins. All of them are subjected to relatively low pressure, whereas in the arterial portion of circulation, the pressure could be up to 120 millimeters of mercury. When we get into the veins, it's only about 10 millimeters of mercury with very little fluctuation. Postcapillary venules are the smallest of the veins, so they're also involved in exchanging fluid with the surrounding tissues. The tunica interna has very few fibroblasts and no muscle fibers at all. Most leukocytes emigrate from the bloodstream through these venule walls into the tissues. The postcapillary venules will combine to form muscular venules. These are up to 1 millimeter in diameter. They have 1 or 2 layers of smooth muscle in the tunica media, and they have a very thin tunica externa. The venules will converge to form medium-sized veins, up to 10 millimeters in diameter. They have a relatively thin tunica media and a thick tunica externa. The tunica interna forms some of these venous valves, which help blood move back up against gravity towards the heart. Blood is pushed up through these valves, and then as it drops back with the force of gravity, it seals the valves. Varicose veins result from failure of these valves and pooling of blood. In addition to the valves, the skeletal muscle has a pumping action which will propel venous blood back towards the heart. As it moves through these valves, if they're intact, they'll hold it in place. We'll also find venous sinuses, veins with especially thin walls and very large lumen, and virtually no smooth muscle tissue at all. There are dural venous sinuses and coronary sinuses on the heart that we've already explored. They're not capable of vasomotion, but they act as collecting areas for venous blood. Finally, we look at large veins. These are the ones that have larger than 10 millimeter or 1 centimeter diameter. They have some smooth muscle in all 3 of the tunics. They have a very thin tunica media with a moderate amount of smooth muscle. Again, the veins don't need to have any pumping action on the blood. The tunica externa, again, is the thickest layer. It contains longitudinal bundles of smooth muscle fibers. The vena cava, the pulmonary veins, the internal jugular veins, and the renal veins are some of the largest veins in the body. So varicose veins result when blood pools in the lower legs. This usually happens in people that stand for long periods and it causes stretching of the veins. The cusps of the valve pull apart as the veins are stretched, and it causes the veins to become enlarged and further weakened. Blood black flows and further distends the vessels, and so the walls grow weak and we develop these characteristic varicose veins. Sometimes there's a hereditary weakness in the veins, but obesity contributes a lot. Often we'll see varicose veins forming during pregnancy. Hemorrhoids are basically varicose veins of the anal canal. Before we complete this section, let's take a look at some circulatory routes. The most common circulatory route is pretty simple. Blood moves from the heart into the arteries, through the arterioles, into the capillaries, then into venules, and then they return back into the veins to the heart. This passes through only one network of capillaries. In a portal system, we'll see that blood flows through two networks of capillaries before returning to the heart. This only happens 3 places in the body. We've already seen the portal system of the hypothalamus and pituitary. We'll see this semester the portal system in the kidneys, and also a portal system between the intestines and the liver called the hepatic portal system. Here's an illustration of a portal system. Blood leaves the heart and moves through the arteries and eventually moves into a capillary bed, which then delivers to another capillary bed before returning venous blood to the heart. Anastomoses are points where 2 blood vessels merge. There are 3 basic types of anastomoses. Arteriovenous anastomoses, or shunts, are where blood flows directly into a vein and bypasses any capillaries at all. Here's an arteriovenous anastomosis. Why would we even have this? Well, often we'll see them occur in the fingers and palms and toes or ears, where they reduce heat loss by not allowing the blood to go out into capillary circulation, which is great for preserving heat, but it can leave us much more susceptible to frostbite. We'll also see venous and arterial anastomoses. Venous anastomoses are where one vein empties directly into another. This provides alternative routes for drainage of a particular organ. This is why vein blockage is much less serious than arterial blockage, because there's often a different way to get there. In arterial anastomoses, seen down here on the right, we'll see 2 arteries merge. This provides alternative routes of blood supply to the tissue. We already examined this in coronary circulation in the heart. Many different pathways to get the blood to the tissue so that in the event of a blocked vessel, this important organ is still supplied. So take a moment here. Close your books and notes and see how much you can remember about the various components of the circulatory system. What's the general structure of a vessel wall? Diagram out some arteries and veins, small, medium, and large. How do precapillary sphincters work? And then what about all the arterial sense organs, the carotid bodies, the aortic bodies, and the baroreceptors we see throughout the circulatory system? In addition, there are 3 types of capillaries that are important for us to understand in order to move forward in the semester. Do what you can to diagram all of these, and then we'll move on to the next section.